That was my second sentence. You're right. It's becoming more and more difficult. Because anything easy has been done. And there are already drugs making money, and asthma, for example. There are really successful drugs that the people are feeling good, but lifelong, when you look at the quality of life, it's getting worse and worse. And the fibrosis, underlying pathogenesis, is not being treated. Now the drugs, new targets, you can forget about the beta-agonists, you can forget about IgE, you can forget. There are so many cytokines, you can forget about all of them. They will not do money, they will not do any good for the patient, because major symptomology has been taken care. You have to go deep into the fibrosis, all this mucus secretion, right? There has to be very different targets. And that's, again, full of unknowns. Who wants to put the money there? The courageous man, the guy, or the culture says, look, I want to go into this. Although the market is saturated, right? We have this argument all the time. Hey, they say, no, I don't want to go into this field. Discover something easy, quick, and all that. There's a tendency for that. But the bottom line comes back always to the disease. How much do you know? Today, we do not know why some people are getting cancer. No idea. There are so many ideas, but we don't really know. Unless you know that, you're not going to have a better drug than the symptom treating drug. Does that satisfy you? Partially? Any questions from the students? In Turkey. Do you want to enter drug discovery after my talk, or do you want to stay away?
I think in your presentation, you did allude on the new concept of personalized medicine and genomics. There seems to be a fair number of
new pharmacology,
new opportunities in this area. And I think any student here or medical student could learn into it a little more. They will find that it's...
You see? Somebody has? Next time. I have slides, but I didn't, I just removed that. Personal medicine is not. Google doctors, they call it. You know that the Google patient has a PhD in that disease area that he or she is sick. And the doctor is just an MD. In the United States, they make this joke. You're talking to a PhD. They know so much. You're right. There are individual differences in pharmacogenetics. I just totally ignore that, except saying young and...
Coupled with that, would you then look at personalized medicine and look at the process of phase 1 and phase 2? So in other words, would you allude on what a phase 1A means, and what a phase 1B means, and what a phase 2A means, and what a phase 2B?
Right. Again, there are a lot of mistakes that have been done. You know, sometimes people have drugs and it didn't work because they just simply did a phase 1. A means just testing the water, a couple of patients just to find out, you know, whether the drug is killing anybody or not, basically. B means sometimes you include some Asian population as if they were normal. So you may see signs whether the drug is effective or not. Okay, so you can divide it into two. This area, I totally get that it's a totally different area, huge, beautiful area. But if you want to know, I have a lot of information, not in this talk, separate talk, at least for the profession, really. I thought this is a general audience, so I didn't go into that. Now, for your information, there are beautiful articles, review articles, suggestions, putting things together, totally new design of phase 1 and phase 2. They are putting the phase 1 actually closer to the preclinical, that it is touching. So you have to discover very early whether the drug has any toxicity in human being, any chance of working, bioavailability and all that. It is new models. When I said again, why is it expensive? You need to change the old mind, the system that I showed you. That has to be changed. And that there are clever companies, especially in the United States, UK, they are trying to do that, but changing the company culture is not easy.
Yeah, changing big pharma culture is difficult.
But small
but there are small companies, what we would call less than big pharma, or less than big pharma. What one can do today, which is very interesting, is that whether it's in silico or whether it's some kind of pseudo-human trials, you can actually predict what you're going to get, because you are not dealing with one target anymore. You are dealing with multi-protein interaction. So you have to look at the overall picture and then decide whether you want to spend $500 million at phase 2, 3/3.
Correct.
So in other words, predict what's going to happen. And today you have the ability to do that.
Correct. I have been offered the job. My wife doesn't know the company because you didn't come in California. Enteros, it's called. They have these beautiful disease models, purely huge computers. Okay? They can tell you if you have a new drug here at the target, the computer knows the disease. Okay? And you say, I want to inhibit this, tell me the consequences. Or I have a chemical like that. I inject into the system. What will be the effects? Interactions with proteins and here and that. It's all happening. In fact, big companies, I don't want to give names, for money, 10 million there, 20 million there, to this company, and I heard there's another one, I don't know the name. But big companies already started using these, especially drug interactions.
Yes, I think what is happening is that big pharma has become a little more interactive with smaller researchers. What they are now doing is they no longer rely on their own R&D. Because most of the people being so large didn't want to make decisions, so they just stayed and kept their jobs.
That is correct.
So big pharma now would go to researchers and they would contract with researchers and say, all right, do this for me, do this work for me.
That is correct.
And that model now is becoming more and more prevalent with big pharma.
Take your mind bias opinion about these companies in silico, it's called, right? They're useful for certain things, but not others. I tell you the limitation. I'm sorry, today I'm seeing the negative sides. Limitation is the following. The computer disease hypothesis, disease knowledge is as good as the literature you put in. Okay? I discovered in this small company, who are the 10 people telling you which paper should we go in? Every one of them is biased. Everyone, I am biased. So computer has the human bias already built in. With the literature and the knowledge that you put in, trustable. So there you go.
So you're ingrained in the bias already.
I wouldn't put them in. I didn't join the company because I didn't really believe. I'm sorry, they're getting the money. I don't know how useful they are. There can be some limited use. Because before giving a drug into a patient, what proteins can it interact is a good question, prediction is good. But these are limited. If the patient takes a hard drug as well as asthmatic drug or blood pressure regulator with this one, what would happen? That's good. I see that. But it doesn't solve my basic problem. I do not know the real cause of a disease. Computer cannot know. I have to know facts. Human being, literature, researcher, whatever you call it.
So before-
That means put more money on basic research.
Thank you very much. Thank you very much again. Another small reminder of Koç University Medical School, Dr. Şefik for the wonderful talk, and we're looking forward to having
Again?
in more sessions in future, whenever he's able. Now that he's spending more time in Turkey, we'll be delighted to have him. Thank you.